Okay, so what I want to do today is talk about specifically neural models, and what I'm going to do is highlight briefly the different types of uh, things that we want a neural model, so that is a neural network model, to have. Other things that biological neural networks have that we don't want our artificial neural networks to have. I'm going to use that introduction to then start to focus on the computational principles that that neural networks implement. And what we're going to do is focus first on the simplest possible artificial neural network. So for those of you that are coming from computer science, this will be in terms of artificial neural network relatively simple, but I really want to focus on what part of the biological mechanisms we need to focus on to start to implement artificial neural networks. So in that sense, conceptually, we're going to see how we build a model, right? What do we leave out, and what do we need to maintain to really keep the core principles alive? And then later on, we can go from this perceptron into more like deep artificial neural networks. And I think at that point, you're going to have enough background, even those of you that have not been educated um, thoroughly in terms of deep neural networks, artificial neural networks, and all of that. You will have the background to read the papers that we're going to read and understand them and be able to work with them. So today I'm going to start out with the simplest of models for how a neuron works, and that's the perceptron. It's a really old model for how neurons work, and where people have, as soon as they understood how a neuron, like a biological neuron, works, they've tried to, find models of how, how can we boil down what that biological neuron does into a model neuron, and the perceptron was one of the first um, models that zoomed in on this type of function that we think the biological neuron implements. So what I'm going to do for today's lecture is go into what biological neurons are doing, and specifically focus on the function that biological neurons implement that the perceptron also implements. I do want to highlight one aspect of this sort of thing that we have biological neurons and artificial neurons, and that is that biological neurons are much more complex than our models, right? And we talked about this before. If you have a model of something, of course, that model is going to be a simplified version of the reality. But I really want to highlight the biological complexity behind this in terms of biological neurons, so that we can better understand what exactly the perceptron tries to focus on. So neurons, we don't necessarily um, think of this, but when neurons were found by Ramon y Cajal, there was a big controversy, big scientific controversy. It's always nice to go back to history and try to understand what exactly people understood at that point in time and what they were totally confused about. Right? So right now we have specific things that we don't really understand how they work. Back then, so this is around the 1900, that decade, there was a controversy where some people thought that the brain is a continuous network, like a fungus has all these, that can be one big sort of coherent organism. Um, and here also people thought that the brain is a continuous network. So there aren't any different cells, but it's one big network. And Ramon y Cajal found that, hey, this brain, if I zoom into it with the stainings that I can perform, it's made up of discrete units. So it's not just one big, jumble of connections of pipes, one big cell. We have these units. They're called neurons, and they make up this network that the brain is. So he was the first to draw out these neurons in their biological detail with the stainings that were there at the time. And here's a drawing of his on the right. You immediately see that there is a very, very distinct structure to these neurons. And of course, in biology, if you see structure, some sort of phenotype, that in many cases gives you very important clues as to the function of, in this case, a cell type. In other cases, the shape of a nose of a bird or whatever. It gives you important clues as to the function of the thing that you're looking at. And that's no different in the case of the neuron here. So what I'm going to talk about today is that this sort of tree-like structure that we see in the biological neuron, that relates to the neuron's function in terms of information processing. And what we're specifically going to focus on is the mapping of inputs to outputs that the neuron implements. And as you see at the left bottom here, this is a sort of schema of a neuron. What you see is that on the left, we have this sort of network arborization of dendrites at the, so that's sort of around the cell body. The soma is what that's called. And inside that cell body, there's a nucleus, right? Because it's, of course, a biological cell. It has to maintain a metabolism. It has to do transcription of genes. And all that machinery to, keep the cell alive is all hosted in the soma. Now, that soma is circled with all these dendrites. And those dendrites are the parts of the cell that receive incoming information. And then as a cell decides to fire an action potential, based on the integration of that incoming information that happens on that dendritic tree, the tree of different dendrites near that soma, it might then decide to fire an action potential, and that action potential is then fired along the axon towards the right, where it then, at the end points, the axon terminals hits other cells on their dendrites mostly, and then those cells can integrate the incoming information from this cell in their dendritic arborization. Yeah, so that's the way the information flows, in this case from the left to the right. And you can immediately see there's a lot of different parts to this structure. And I want to first, in this lecture, dive into the intricate structure of the biological neuron. Not too deeply, because this is a rabbit hole. We could spend months talking about exactly what the cell does in terms of how it integrates information in a biological sense. But I want to give you an idea of the biological complexity that we then boil down to create our model neuron, the perceptron. So if we look at how the neurons function as a neural network inside the brain, they are all connected. So they're forming a network, right? So the input of one neuron is the output of another, and so forth and so forth. So they're all connected, and they form a network. And they're not like one big sort of circle cluster of different cells, but they're usually, in most brain structures, organized such that they are segregated into different levels, different layers, and that information flows through different levels. So from one sort of network to the next network to the next subnetwork, and so forth and so forth. So what you see here on the right is a diagram of the visual system. On the bottom, it says RGC, which stands for retinal ganglion cell. That then projects its visual information, the retina that is, to the thalamus, where you see LGN, which stands for lateral geniculate nucleus. I'm just giving you a bunch more information. This will not be in the exam. Um, but then the information is projected into the cerebral cortex, and there you see this sort of expanse of a network arises. We start with these specific parts where, for example, we have V1, V2 at the bottom. Those are really the basic sort of areas of the visual system. And then information is projected up to these subsequent layers, and there you see that they're all intertwined. And this is something that I want to really highlight here. We have this hierarchy of many, many different visual regions. And although it's true that the lower levels are closer, in this case, in this example case of the visual system, that the lower levels are closer to the retina, and higher levels are closer to movement output or memory or planning. We have this sort of low level and high level progression. That's definitely the case, and there are different levels in between. I want to highlight here that we think of information flowing from the bottom to the top. Oopsie. Oh, this transition takes a long time. Now I have to wait. So we think that information flows from the retina all the way up, and that's the way we generally think about these networks if we want to simplify them. But it's really important to realize that what you're seeing there, for example, right in the middle of that diagram of the human visual system, or the macaque visual system in this case, there's a lot of feedback, and this is really a tangle of communication between these different areas. So one of the steps that we'll take if we're going to take this biological system and boil its principles down is that we're going to focus on that feed-forward sweep of information that that animation highlighted. So we're going to pretend that we're just going to stimulate the retina once, and then information will flow in this bottom-up way, and that's it. And that's the first simplification that we'll perform. And it's already immediately important to realize that that is a humongous simplification, right? The brain is filled with feedback processes and lateral processes that are, in general, in the simplest neural network models, artificial neural network models, these types of connections are usually, just thought to not exist. And then we are going to see how far we get with that. So that's the idea. Right? We have a neural network that's connected. It has layers. These definitely exist. And for our artificial neurons, we'll think about this being just a bottom-up sweep, and that there is no more complex communication between these different layers, although in the biological system that definitely does exist. So if we're going to create artificial intelligence, right, we're looking at this biological sort of structure. What do we need to do, right? This first part of the lecture, I'm really going to focus on, if we want to make artificial intelligence based on the principles of biological intelligence, what do we need to focus on, right? So the idea is we have this real intelligence architecture in the brain, and can we try to copy this to make information processing systems that are artificial? Now, neurons are really, really complicated. They have more complex connectivity, like I just highlighted, than the simplest artificial neural networks. But what these two movies here show is two elements that I think are very, very important to focus on, and to really understand that they happen in a biological brain. So the top one, the light gray one, you see neurons in a culture just changing the way that they interact with one another. They're really changing their network structure. You really see new connections forming all the time and others being broken down. And this happens in a very sort of biological, which means in this case, messy way. Right? There's a lot of noise in there, and these cells are all doing their little thing, trying to find whether their neighbors are doing similar things and all of that. So what you also see here on the in the darker image, so this is a movie of a calcium imaging in the mouse brain, where you can just point a microscope at a specific piece of tissue. And here you see all of these neurons are more or less organized in the same direction. And that's what you see there. Um, and so the calcium imaging, what it allows the researchers to do is see calcium concentrations inside the neurons. And as a cell fires, its calcium concentration changes rapidly, and that's what you're seeing. So what you're actually seeing is these cells are firing action potentials in the tissue. And what you see is time is of the essence, right? You see waves and clusters of sparkles of activation flowing through this network. And this is something that we don't have if we are building an artificial neural network that just has single activations, right? So that here you see the time is of the essence at different time scales. So in terms of the activations in the darker movie, and in terms of the connectivity and really the structure of the whole network, as you see at the top there. Now, what we want to do is get a good model, right? We want to simplify the situation and still retain the basic characteristics. But as we're creating this model, we need to be mindful of the fact that we are simplifying things, and we need to understand what we're throwing away. Because if at some level our artificial model turns out to be not functioning the way we want to, maybe we can try to plug in some of that biological stuff that we threw out before in order to recreate some naturalism, to add some functionality. So what are we going to throw away? Well, first let's just look at how we're going to. So what is it in terms of these biological neurons that we can throw away? So biological neurons are like really varied. They have different shapes. Um, they have different arborizations in terms of the axons. So here at the top is the axons here. And you see that there's a lot of variability in how exactly these neurons are shaped. This is a standard excitatory neuron. So when it fires, it excites the subsequent cells. Here's the dendrites, and here you see the axon. It's a very large arborization of the axons. Here, for example, you see a Purkinje cell in the cerebellum, and these have these huge trees attached to them. And these are planar. So this looks like it's a 3D thing. These are actually very flat if you rotate them. And then you have very tiny cells. Now, these cells can be excitatory in terms of that they excite the cells that they project onto, or they can be inhibitory, that they inhibit the activations of the cells that they impinge on. But in all of these cases, we see this basic structure reappear. That is that of dendrites, a cell body, and an axon. So let's have a brief look at this, how this schematic of the neuron, how that is complex in terms of the functions of the dendrites and the axon, for example, and how we can then start to boil that down. So on the left here, you see a somewhat complex figure on the different parts of a neuron. And I'm not going to quiz you about all of this, but I thought I'd add this just to show you how many different moving parts there are to the biological neuron. And what we're going to focus on right here is not just all of this, but these specific parts where the dendrites are. We're going to focus on those dendrites and how they integrate information. Because as we've already seen, we have this input to output relation that we are going to focus on. These are the input side of things. As we go to the output, if you see on the right, this integration that happens in this dendritic tree will at some point lead to an action potential that then travels across the axon and then fires this action potential at the subsequent, projects this activation to the subsequent layers of neurons. But it's important here, this input-output segmentation, to realize the signals, they arrive in the dendrites. And as I just already said, as these signals come in, these action potentials come in from the previous layer onto these dendrites, they can be excitatory or inhibitory, which means that in terms of the currents that are generated, they can be excitatory. So these are called excitatory postsynaptic potentials, or EPSPs, or they can be inhibitory postsynaptic potentials, or IPSPs. Now, what does that look like? In a very simple schematic, and then I'm going to repeat this type of schematic. So let me just explain what it looks like, what the different parts are. So here we see soma, and here are some dendrites. And if you see a circle going onto that dendrite, that's going to reflect an excitatory synapse onto that dendrite. Now, as this synapse fires, right, and it releases the neurotransmitter there, it has, of course, an effect on the dendrite here. What happens is some current starts to flow as ion channels open. I'm not going to go into all of that processing because I think it's a bit too low a level for us to be interesting. But then what you see is that there's going to be this excitatory postsynaptic potential. So that just means that the voltage, the membrane voltage changes. Now, what type of mechanisms are there as these EPSPs and IPSPs are sort of thunderstorming onto this dendritic tree? What are the dendritic mechanisms that make the neuron either fire or not fire? So one important thing to realize is that if multiple excitatory synapses fire at the same time, or are being activated at the same time, these EPSPs will combine across space. So they will sum together. As you see here, the membrane voltage will, the deviation of that membrane voltage will increase as multiple of these EPSPs arrive at the same time at different locations. And of course, at some point, the cell will perhaps start to fire if this membrane voltage crosses a certain threshold. Now, that's the idea. So this processing here, we see that there's this combination across space if things happen simultaneously. At the same time, these EPSPs also combine across time. Right? This potential deflection that we see takes a prolonged period of time. So if in that same interval we get a new synaptic activation, that will create another membrane potential or membrane channel opening, ion channel opening, and it will create this new potential change. And these will combine across time. So if enough incoming signals arrive in a very, very brief period of time, we will get this membrane potential change to combine, to accumulate, and then maybe lead to the firing of an action potential. Now, it's important also to realize, and this is all how biological neurons, again, are more complex than artificial neurons, right? In many cases. So one important characteristic is that here I'm showing you this entire sort of dendritic tree, which has, in this case, only 2 branches, but still. if you record the voltages, the potentials at different locations on this neuron, if we get very strong EPSPs arriving at the most distant parts of this arborization, so that's around here, there we see strong voltage changes, but that dissipates, and as you go towards the axon here, that change is less. Yeah? So there's a gradual decrease of these potential changes from these dendrites to the soma, where the neuron decides to initiate an action potential if it does so. So you can imagine that this is a very, very strong oversimplification, right? You have this enormous dendritic tree, and you get all of this stuff happening, the EPSPs coming in, combining across space at different junctures. And of course, it then matters how distant these different EPSP incoming points are. And they also combine across time. So what I want that to illustrate is how much computation is actually already happening inside the dendritic tree. You can see how much potential there is for information combination in that dendritic tree just by virtue of these simple mechanisms. Now, one other extra simple mechanism here is that if we have excitatory and inhibitory postsynaptic potentials in that dendritic tree, we get these processes where inhibitory postsynaptic potentials can cancel excitatory postsynaptic potential. So you can imagine if you have an excitatory postsynaptic potential here on this dendrite, it will create this potential change that will be flowing from the dendrite towards the soma. And that's what you see here in this column. If you then have an inhibitory postsynaptic potential somewhere as this potential is flowing towards the soma, what you see is that can cancel out that excitatory potential completely. Yeah? So not only do you have this complex set of different interactions that can happen in the dendritic tree by virtue of combinations of excitatory postsynaptic potentials, you also have all these inhibitory postsynaptic potentials that can sort of shape the flow of information in the dendritic tree. So you can imagine this already gives just a single cell has up to something like 20,000 different synapses impinging on it, right? And these are in a continuous sort of orchestra of excitation and inhibition, where that single cell goes through incredibly complicated decision machinery to decide whether it's going to fire an action potential or not. And every neuron in the brain has this type of processing going on. Incredibly complex. But of course, we want to go to model neurons, artificial, simplified model neurons. And so all of this is beautiful, but how can we start to see what the basic computational principle is that the neuron implements? And if you go into how people have done this in the past, people have focused on this basic element of the input-output transform that the neurons implement. So that is, if this is our schematic of the neuron, and we're just going to disregard much of the incredibly complex machinery in the dendritic arborization, we're just going to say, okay, this is our model. So what we're going to do is we're going to say this model is a unit, or this model neuron is a unit that integrates a whole bunch of inputs and then projects out. So you can see how this is an oversimplification. So we're going to throw away the temporal dynamics of the integration in the dendritic tree. So that's all gone. All of the dynamics in terms of ion channels and voltages that might interact, these are gone. What's also gone is just the spatial complexity of the dendritic tree, right? Having distant dendrites and having more close to the soma dendrites. All of that is gone. And we're going to model this just in terms of the most basic input-output transform that each of the units is going to perform. So this leads us to the perceptron. So those of you that have worked with AI things before, how many of you have implemented a perceptron yourselves? You can just raise hands with a Zoom function. I just want to see some hands. Not that many. Great. Or I'm missing a lot of hands. That could also be. Oh, yeah. Great. Okay, so we are going to, in the practicals, implement our own perceptrons. So then that'll give us a little sort of playground to work with to really zoom in on this basic computational principle of the perceptron. But what is that? I already said this is the input-output relationship, but let's dive into what that perceptron does. So it's similar to neuron in that it transforms inputs to outputs. And I already showed you this very, very sort of simple schematic of what it does. This is what we can, this is, again, the same schematic, but a bit more exploded. So this is the simplest version of the perceptron. We're going to go into a slightly more complex version of the perceptron, but that's for later. So this is that input-output relationship schematic, making that biological function, boiling it down to the bare necessities. So how does this work? Well, this model on the left here takes inputs. Right? We're just going to take this left or right flow of information as the standard. And this model takes a real valued input. So it's like floating point numbers, continuous numbers on a set of different inputs. Right? So there's a vector of inputs. It goes from x0 to xn, depending on how many input channels there are. So you get this input. Now that's great. That can be anything, and we'll go into what that can be later on. Now, each of the inputs here is scaled by a weight that is similar to the strength of each of the synapses in the tree of a biological neuron. Right? So some synapses can be strong, others are weak. And of course, in this dendritic arborization, the neuron can scale the different weights of these synapses, and that's exactly what we're going to do in our model neuron, in our artificial neuron, in our perceptron. So this is just how much is each of those input numbers scaled in that integration process that the neuron performs. So these are similar to synaptic weights of the biological neuron. Now, what this model neuron does is it integrates the inputs. So similar to this very complex dynamic type of thing that happens in the biological neuron, that sort of integrates all the information for the neuron to decide whether it's going to fire. Here, that is simplified to a dot product. So that means we just in place multiply all of the weights with all of the inputs and then sum them. So that's what our neuron does, or our neuron does. In this case, a very simple linear summation type of processing. And the advantage for us is that this type of computation is very easy and fast in computers. So this type of thing is exceedingly fast for a computer to perform. Now, we're almost halfway here in terms of the diagram of what the perceptron is doing, because the next step is to pass this sum, this weighted sum of all of the inputs, through an activation function. And we can think of that as the decision to either fire or not. And this decision to fire or not depends then on a threshold that we can set. So if the activation, that is this dot product, is larger than some threshold theta, it's going to fire this neuron. So it's going to have an output of 1, and it's going to have an output of -1 or 0 otherwise. Yeah, so there's these different parts of what the perceptron does is we have settable weights. So we have a vector of weights that can be set. We have this summation of the multiplication of these weights with the inputs, and these are then summed. Then that sum, that single number, is compared to some threshold that determines whether that artificial neuron will fire or not. So these are the different sort of stages of what the perceptron is doing. And you see that this is incredibly simplified relative to the biological neuron, right? So the biological neuron has weights that depend on metabolism timescales on different types of processes, right? You can have electrical integration of information at a brief time scale. You can have that proteins are synthesized and brought to specific synapses that increases their weights. So this is all much more complicated. That summation process is also much more complicated because it requires this integration of changing voltages towards the soma of the neuron, and then it decides to fire, which just puts forth a single action potential in the case of this decision to fire. So that's quite different, right? There is no time in this model, and there's just activation versus no activation. But that is trying to capture, right? We're leaving a lot out. That's completely clear. But what we are retaining is this sort of taking up a bunch of inputs, weighting them in some way, and then creating a decision based on that weighted input pattern. So that's our model of the neuron. So we've just boiled down all of that biological complexity to this single mechanism. And then the question is, what does it do, right? This model neuron is a little classifier, right? If it fires 1, if there is this threshold, if we're going over a certain threshold, and 0 if we don't, then what this model neuron does, it classifies whether some pattern of inputs is present, right? That's in essence what it does. And this decision is based, of course, on the weights that we've set for this perceptron, because those weights determine exactly what pattern it searches for to base that decision on. And in this sense, even though we've boiled down a lot of that biological complexity, it is very similar to what actual neurons do. And for example, in terms of sensory systems, neurons will fire only when there is a specific pattern of inputs. So on the top here, you see an orientation selective neuron in the visual system. And you see that if we have an input where this bar, so here on the left column, you see the inputs, and here you see the spike pattern. So this is really whether it's firing any action potentials. If we change the orientation of that input, we see that first this model of this neuron is silent, and then it starts firing and then goes down again. So this progression of orientation we can plot on this axis here. Just center it on 0, and we see that the firing rate of this neuron depends on the specific orientation of the input. Yeah, so in this case, the pattern that the biological neuron is searching for or is responding for is some orientation in the input, and you can imagine that that's just resulting from some weighting of further inputs. Now, similarly, these neurons, they can have a spatially well-defined receptive field. So this this specific neuron has a receptive field where it fires when there's something at this location in the visual field, and it inhibits its activation as something is in this portion of the visual field. So this again here is the sensory input pattern that makes this neuron fire. Yeah. So you see that this pattern recognition part of the perceptron is very much biologically inspired. And even though we've taken out all of this biological detail, this is still the function that it's performing here. So